Blackboard, okay? Something black. Okay, so this is the overview for the flow of genetic information. This information of content of genes is in the form of the sequence of nucleotides in the DNA. So you have your DNA, sequence of this DNA that will have for these genes, and these genes are going to be expressed, gene expressions. From DNA will go for RNA transcription, and then from RNA will go to like the amino acids, polypeptides, proteins, enzymes, translation. Okay, that is the gene expression. These proteins are the links between the genotype and the phenotype. Remember that example about like the Miriam's nose she got from her mom? That is not true. It is the alleles, right? So that's what it got, the genotype, and then once they get expressed, phenotype. So proteins are this link between the genotype and the phenotype. DNA is inherited by an organism, and that leads to specific traits by the synthesis of those proteins. What alleles are going to be transmitted from the parents to the offspring. So that is the gene expression. Make sure you know this. By this question, for sure, it will be there on the exam. What is gene expression? The process by which DNA directs this protein synthesis, includes transcription and translation. So here you have, the wild type, phenotype, and then the alterations or alternative phenotype, the mutant, right? Once it is not the wild type, okay? Any particular mutation that is after alterations from the normal wild type. So these alterations in the sequence of a single gene, then it can have these profound effects on the organism. For having a different color eyes, different color on the mice, extra digit on the hands and feet, or just by a mutation. This is what is the cause. Okay? And like different shapes, you can see here from the leaves. So the wild type, that is the normal phenotype, characteristic that exists naturally. The mutant, that is the organism that is displaying the new phenotype. That is not found in wild type organism. Okay? So that would be mutation. So how was the fundamental relationship between genes and proteins discovered? This whole gene expression that we have been talking about, transcription, translation. How was this discovered? So it began with this scientific inquiry with George Battle and Edward Taylor. They exposed this bread mold to X-rays, and that created these mutants that they were not able to synthesize essential amino acids, arginine, for example, for them, on minimal media. Okay? So you have complete media and minimal media. Minimal media doesn't have arginine. Okay? So these organisms, this bread mold, they need arginine in order to grow and to develop. Okay? Complete media has it, you put them in there, they will grow. Minimal media does not have arginine, so you put the wild type bread mold, it will grow in there because it will take from this minimal media and it will convert that into the arginine. It will be able to grow. But if you place this bread mold that has been exposed to X-rays, now there has been some changes on the DNA, some mutations. Then if you place this mutated bread mold on this minimal media, they are not able to grow because they are not able to convert that minimal media precursors into the arginine that is necessary to grow. Okay, so something happened whenever they expose this bread mold to the X-rays, to the genes, to the enzymes, and then that affected their survival. Okay, so using this process, they and their co-workers, as SRB and Horowitz, they identify these three classes of arginine deficient mutants. So each lacking a different enzyme that is necessary to synthesize arginine. Okay? From that, it was developed the one gene, one enzyme hypothesis. In other words, that the X-ray mutated one gene, and therefore the enzyme that was coded in that gene was no longer functional. One gene, one enzyme hypothesis. Okay? So that's basically what it says. That you state that each gene detects the production of a specific enzyme. So that's what you have right here. You have the bread mold, the neurospora cells, okay? So they have these, they place them in complete media, so they're able to grow, okay? Complete media, able to grow. Then they expose them to X-rays. It will change their DNA. Now, the nice thing here, it's really important to know that these organisms, neurospora cells, this bread mold, they are haploid, not diploid. Remember, whenever Mendel did experiments with the pea plants. Well, they are diploid, right? It has like two alleles, it's a big P, little p, right? This one, that's not the case. These ones are haploid. So they only have one set of genes, okay? So that means if with that X-ray you mutate that one, you're done. That gene is done. It's not that it has two alleles. Oh, maybe you just like affected one, but still this the other one, right? No, in this case, you affect one gene, that's it. You can see right away the effect that you will have once you mutate that gene, okay? Really key. Haploid cells. All right, so you take these neurosporal cells, subject it to the X-rays, mutation, boom. Then you put them here, these surviving cells, they will form like these colonies of identical, genetically identical cells, okay? Because they are born. You see those colonies, but they're like millions of cells, okay? So these have been mutated, okay? But once you remove them from this complete media, they're able to grow there because they have the arginine red, right? Complete media has the arginine, so they don't have to worry about that. But once you take those mutants and put them on minimal media, they don't grow, they die. Why? Because something happened over here that the X-ray mutated these colonies, these organisms, and therefore the enzyme that is necessary to convert this minimal media into the arginine is no longer working, no longer functional. So they are not able to live, okay? If you put this one, the control, that is the wild type, no X-rays, no mutation, you put them in minimal media, they grow. They're able to convert that minimal media into the arginine and therefore survive, okay? If you put these mutants right here in the minimal media, but now, even though they are mutant, they don't have like a functional enzyme, but now if you put additional nutrients, the one that is lacking, the one that the enzyme is no longer working, they're supposed to do, they're supposed to produce, if you add that, the product of that faulty enzyme now is able to grow. You bypass the need of that particular faulty enzyme because now you're adding the product of it, okay? You're able to escape that. You're no longer, that organism is no longer going to die because of that faulty enzyme. Like, I don't need that faulty enzyme, I have the nutrient that faulty enzyme is supposed to produce. Okay, if you add that nutrient in the media for these mutant cells, they grow. That's how they develop this one gene, one enzyme hypothesis. So that's what they did. They did all these experiments, and for example, like one set of mutants called arginine for growth, and it was determined these different classes of these mutants were blocked at different steps in this biochemical pathway for organ synthesis, and that's what they have this right here. So, if the X-rays mutated gene A, enzyme A is a faulty enzyme. Okay? So, if the X-ray mutated here, enzyme A no longer working. They put this organism, neurosporal cells that have been affected by the exposed to the X-rays, they put them in the minimal media, it doesn't grow. Why? Because now enzyme A cannot take this precursion in the minimal media and convert it to ornithine, which then enzyme B will make it to citrulline, and then enzyme C will make it to arginine, and therefore the organism is able to live. The goal is to make this so the organism can live. Okay? But now if the mutation affects this, they cannot make ornithine anymore and live. Okay? So the goal here will be put this mutant cell in the minimal media and add ornithine, and now it's able to grow because now you're adding the product of the enzyme A, and now you add ornithine, enzyme B is working, can make citrulline, enzyme C is working, citrulline can make arginine, and the organism lives. Okay, that's how you define it. Now, if the X-rays affected gene B, now enzyme B is no longer working, right? If you play this organism in the minimal media, it will not be able to live. Why? Because enzyme C is working, it makes ornithine, but now enzyme B is not working. It cannot make citrulline, right? And therefore, it will not be able to make arginine. Therefore, it dies. If you add this organism to the minimal media and ornithine, still, it will not be able to live. The problem was not enzyme A, the problem is enzyme B. So in order to bypass this, you add citrulline. If you add citrulline, you bypass this, right? You add the product that the faulty enzyme is supposed to make, and now enzyme C can turn this into arginine, and the organism lives. Okay? Now, if the X-ray affected gene C, now enzyme C is faulty. It's not able to work. Okay? If you put this organism that it has a faulty enzyme C, put the minimal media, it would not grow. If you put the minimal media not ornithine, it would not grow, because the problem is not enzyme A. If you put this one minimal media not citrulline, it will not grow, because the problem is not enzyme B. You have to add arginine to bypass the need for the faulty enzyme C. So that's how you determine which enzyme is the one that is faulty. Okay? You will put the mutant cells, put them in minimal media, and then start adding ornithine. Okay? If it grows, then enzyme EA is the one that is faulty. Right? If you add these, or if you add this and doesn't grow, right? So if it grows, that is enzyme A, right? If it doesn't grow, then add citrulline. And now if it grows, then that is enzyme B, the one that is not working.
If ornithine doesn't grow, that means it is enzyme A.
What's that?
If ornithine doesn't grow, ornithine, whatever that is, if it doesn't grow, then enzyme A, right?
If ornithine, if you add ornithine, let me put it this way, okay? We have the black one? So if you add ornithine, right? And it leaves, then that tells you that it is enzyme A, the one that is faulty. Okay? If you add ornithine, but it doesn't leave, then that tells you that enzyme A is not the problem. Enzyme A is working. Okay? If you add citrulline and it leaves, then that tells you it was enzyme B. Okay?
Are you saying if it leaves?
It's if grow, leaves, it's able to grow.
Grow, okay.
It's alive.
Okay. Lives.
Leaves.
Oh, lives,
Yeah.
okay.
If it alive, okay?
Okay.
If it is alive,
Okay.
if it grows, okay?
Okay.
Then it will tell you that it's enzyme B, the one that is not working. If you add citrulline, but it doesn't grow, then you know that this one is not the problem. Okay? It will tell you that this one is the problem. So then if you add arginine, right, and it grows, and that tells you that it's enzyme C, the one that is faulty. Okay? Does that make sense? Okay, again.
No, not again. Is that part that you confused? He said if it grows, it's not the one.
Okay, can I try something?
Yeah.
Okay. So enzyme A, it synthesizes the orthanine from the precursor, right?
But yeah, this one is making this.
Okay, and so if it has the precursor and orthanine is not created, enzyme A is broken.
Wait, can you repeat that one?
Okay, so if you add the precursor and orthanine does not get synthesized or created, enzyme A is malfunctioning.
It's not functioning, that is the problem, yes.
Okay. And so then you add orthanine and then it grows.
Excuse me, sorry, guys? They couldn't hear what they're saying. Do you guys hear?
Oh, yes, thank you for addressing that. Yes, yes, so everyone, if, yeah, let's do this together. Were you having an answer question about this?
I was just explaining.
Okay, okay. Right, so what was the question we hear?
Nobody asked question. I was
All right, so let's do this together so everybody can listen. Yeah. All right, so let me do one time and then one more time and then let's see, okay? For everybody who has a question. So this is minimal media, all right? Let me put some diagrams. Minimal media, that's the precursor, okay? wild type. Okay, it has enzyme A. Enzyme A will take this precursor mineral media and make ornithine. Once you have this ornithine, enzyme B will take it and make citrulline. Okay, once you have citrulline, enzyme C will make arginine, and therefore the organism is able to live, grow, develop. Okay, will grow. Okay. If enzyme A is not working because gene A is mutated, right? So it cannot make this process. So in order to bypass this, you add ornithine. And now it's able to continue. Okay? So how do you know that the enzyme that is faulty is enzyme A? Grab the mutant cells, put in the immuno media, add ornithine, and if it's able to grow and be happy, then you know that the problem was enzyme A. Okay? If you put minimal media, the enzymes will not grow, and you add ornithine, right? But it still doesn't leave. Okay, so you just do this right here. The same thing. Add ornithine, but it doesn't leave, but dies, doesn't grow. So that tells you that the enzyme A is actually working. That's not the problem, because now you added ornithine and it still dies. Let me just put the ornithine because it looks like another face in there. Right? Enzyme A is not the problem. Enzyme A is working. Okay? So what do? Okay, so now you add to this dying organism, you add citrulline. And now with this, it lives, it's happy, it's growing. Then that tells you that the problem was enzyme B, the one that is not working. Okay? This right here, this tells you that enzyme B is working, enzyme C is working because able to complete the pattern. Here, this tells you that the enzyme C is working because able to complete the pattern. Okay? Now, if you add ornithine and leaves, it grows, so that tells you enzyme A is working. If you add citrulline and it doesn't grow, it dies, so that tells you that enzyme B is working. That's not the problem, right? So what you do is add right here arginine, and then it's able to live and grow. So that tells you that the enzyme C was the one that it wasn't working. Does that make sense? Is it better?
Mm-hmm.
Kinda? Who's, yes?
The third line example, shouldn't it also be a sad face, like dying, if you add the ornithine on the third line? Yeah.
This
Because
one?
if it's smiling, that means that the enzyme A
Oh,
is
sorry.
the one that's
Yeah,
working.
it's good.
He should be dead.
Yeah, should be.
Yeah.
Thank you.
You should be dead.
I know my body.
Yeah. Yeah, so you add the ornithine, it doesn't grow, right? I did say that grow, but I don't know why people use funny face. It doesn't grow, so that tells you enzyme A is working. You add citrulline, if it doesn't grow, that tells you enzyme B is working, right? So now you add arginine, and it's going to live, it's going to grow. It's going to be happy, okay? Thank you for catching on. Does that make sense? Yes? Who still says like, no, because you will see like 2 questions about this on the exam. Final exam and exam.
Okay, I'm
Exam,
confused.
good.
Okay. So enzyme A, so on the second row, it said, so enzyme A, it's fine, right? Okay. Because you add ornithine and it doesn't grow.
Mm-hmm.
But it does grow if you add citrulline. So you know it's enzyme B.
Mm-hmm.
And then for the one below it,
So you add ornithine, still doesn't grow, right? So that tells you enzyme A.
okay, I get it. Yeah, cool.
You add citrulline, and it still doesn't grow,
Yeah.
right? Tells you enzyme B is working. So now you need to add arginine, and now it
Okay.
grows. That tells you that enzyme C is working. Okay. Question? Oh.
And if we add arginine, and it still doesn't grow, or does it grow or does it not grow?
Arginine? Yes, because this one is what it needs to grow. That will be an essential amino acid for them in order to survive.
So basically you're saying C is always a healthy enzyme.
It's not always the healthy enzyme. So here, this one is the one that is faulty. It's not making arginine. So you adding the product of itself to bypass the need of enzyme C. You add the last product, as it were. Okay. All right. So let's do this analogy. Right. I need like 3 volunteers.
Come on, because I'm falling.
I don't want
3 volunteers.
Oh, is it like insulin? Like if you can't make insulin,
That's right. Then
then you die if eventually if you just can't make insulin at all and you eat sugar, right? And so like you get like fake insulin. You get fake insulin and then you live.
You are in bypassing the need for that one. Yes.
Yeah.
All right. So let's add you over here. Let's add you at the times. All right. So here we have over here a little more closer. Enzyme A, enzyme B, enzyme C. All right. So I'm going to add. Yes, thank you. Okay, so this is a precursor, okay? Minimum media. Precursor, right here, okay?
What is that again?
That's a staple. That's it. The precursor, okay? Precursor to the amino acid. Precursor to the amino acid, okay? Amino acid precursor. We have here the precursor. Here, this one is ornithine, okay? Remember this. Ornithine, citrulline, arginine, okay? So, wild type. We put the wild type organism, bread mold, your spore cells, immuno media right here. You will take this and it will convert it to this. So you will grab this and say, okay, I'm enzyme A, I grab the precursor, I make ornithine, okay? You make ornithine. You grab this and make this. Once you have it here, then she will say, I'm enzyme B, I take ornithine, she will take it from here, and it will say, I make citrulline, okay? Then enzyme C will say, I'm enzyme C, I will take citrulline, and I will make arginine.
We ain't never had talked this much before.
Once it makes arginine, it's able to grow and live, okay? All right, so let's do the wild type. So you will need this, don't show it. Just once you grab this, then you show that, okay? You are going to make this, you're going to make this. Don't show it right now, until I give you this, okay? So let's start this chain of reaction, okay? Precursor, I put you right there. What are you going to do?
I'm taking.
Go ahead, say it.
I'm taking.
Who are you and what are you making?
I'm enzyme A and I'm making ornithine.
You take the precursor and you make ornithine. There you go, okay?
And do I give this to her?
No, that's it.
No.
I'm enzyme B and I make citrulline.
From
I
what?
keep this
From what?
from enzyme B.
You make from ornithine, you make citrulline.
Oh, from ornithine I make citrulline.
Yes. So you hide that one and you give them that. No, you hide this one.
Okay.
Because what you're doing is this, can you see? You're like,
Consuming it?
yes, you're enzyme A, and you grab ornithine, and I make citrulline, okay? So you converted this, okay? And then?
I'm enzyme C. I take citrulline and I make arginine.
Arginine, perfect. And now it's able to grow. Right, let's go back. You got this, okay. So now, let's say that I'm adding this neurosporal cell, I expose them to X-rays, and I mutate gene A. So now she's no longer working. Can you face the board? Okay. She's no longer working, okay? I place this organism here, over there. She's no longer working, right? Yeah, that's, yeah. All right, face this way.
Just,
Face-
yeah.
No, face me, face me. There you go. She's no longer working, right? So now she's no longer part of the whole biochemical pathways. Okay? So I put it in minimum media. She's not going to see this. She's not going to grab this because she's not working anymore. Okay? So,
Nothing works.
so they don't work.
Okay.
Why? Because she's not grabbing this and converting that into ornithine. Right? So she's like, I'm ready to go. I'm ready to make citrulline. Where is my ornithine? She's not working. Right? So what do? They die. Exactly. So how can we make this organism live or grow?
We add her even though she's not there, right?
We don't add her. We add what she makes. Yes.
So I'm dead.
You are not working. Yes. So we are ornithine. So I bypass her. Okay. I bypass the need for her, and I
Who
say,
adds it?
Hmm?
Who adds it?
the researchers. We add the ornithine. Okay. We add the ingredient that she's supposed to be making. Okay. So I put this organism. She's not working. She's not converting this into ornithine. But I put you in minimum media, and I add the ingredient ornithine. So what do you do now?
I take the added ornithine and I make citrulline and I pass this to him. I hide
And
this.
take citrulline and
Sorry,
make it to
guys, that was
arginine.
And now they live. Now they grow, right? Okay, let's go back. Give those things back. Okay. So now enzyme B is not working.
Yay.
Face here or the back or there you go. Face whichever side you go. Yeah. Okay, good. Now this one organism is supposed to X-ray, right? Now gene B mutation, enzyme B faulty enzyme not working. I place this organism minimum media. What do they do?
I take,
Okay, say it.
wait,
Mhm.
do I take
Okay,
it?
sure. Mhm.
So I take the cursor and then I make ornithine
And then what happens?
and someone brings citrulline.
No, wait. What happens here? What happens here? So what happens? You make the?
She ain't working.
She ain't working. I mean, she ain't
Yeah.
work. She's working, but she's not working, right?
Yeah.
So hide that one. There. She's not working. So what happens there?
Someone brings.
No. What happens here? For the cell.
It just stops working.
We're dead.
You're dead.
Dead. It dies, right? So what do we need to do in order to survive, for this cell to survive?
You take enzyme A
You
and put it
take
in
the,
Hmm?
You add
Citrulline.
Wait, it's
Citrulline.
We add what?
citrulline.
Citrulline, because that citrulline is what?
Is enzyme B making?
The product of enzyme B, right? So I bypass the need of her and I just add this citrulline, and what happens here?
It lives.
It lives.
you make iron.
It lives, it grows, survives, right?
What happens to mine?
It's there, right? Enzyme B is not utilizing that one.
Would
So
it
it
have a
just-
buildup of ornithine or not? So
Well,
she's
it-
working,
She will just like reach saturation, but that's it.
she would what?
Like reach saturation, it will just take whatever precursor is there and make the ornithine, and once it reaches equilibrium, then that's it.
Ah.
And enzyme B is not working, it will not, even though there is increased concentration of the ornithine, it will not do anything. And here you have like enzyme C, like I'm waiting for my citrulline, but she's not working, it will die. Until you add the citrulline that is supposed to be making the enzyme B. Okay? What happens if you place this organism, minimum media, and you add ornithine? What will happen?
There's
It
too
would
much.
die.
It still will die because not working, right? Enzyme C will not be able to make that into the arginine, right? So that's why basically what is happening over here. You add extra ornithine, it will still die because enzyme A was not the problem. The problem is enzyme B. It doesn't matter how much you add of the ornithine of the substrate, enzyme B will not use it. Okay?
So I'm gonna be dead, but he's gonna be fine.
Well, this is the enzymes, right? The organism is the one that is going to be dead or alive. Okay? You're going to be working, you're going to be functional, but she's the one that is not functional.
Yeah, but over there you have dead.
This one is the red blood cells. Okay? What happens to the organism? Yeah. So here, the organism dies because enzyme B.
So once it's added, am I still dead?
Remember, you are the protein, the organism, right? If you add citrulline, then the organism lives or grows, survives.
But the enzyme
the enzyme. But if the organism dies, forget the protein. Okay? But I think about you as part of the pathway, and the whole room is a cell. Okay?
Okay.
If the cell doesn't make this, the cell dies, and that's the sad phase. Okay? If your tree, the pathway makes the arginine, the black one, the cell leaves, happy phase. Okay? So seeing yourself as part of the pathway to make
So
happy
like a
phase.
limb?
Mhm.
I have a question.
Yes.
Okay, so she's dead, sorry. She's dead.
Not functional. Right, remember, whenever you use the proteins, these enzymes refer to them as functional or not functional.
Okay.
And if they have these, then the organism, okay, they are proteins of the organism. If they have these, the organism, the cell lives, grows. If not, they die. Okay?
So she's not functional. Who's gonna transfer who's gonna give her,
The researchers added.
well, since enzyme B is the one transferring to adenine, to make adenine, who's gonna transfer the thing to?
No one. The researchers, they add the citrulline,
They
right?
add the citrus, but she doesn't do anything. She just keeps making. What is it?
Just
Or-
giving
or-
ornithine.
yeah.
Ornithine. She just make ornithine, right? You are not going to add the enzyme. Right? If you were going to add a functional enzyme, then that functional enzyme would take this into that,
So
right?
you need the enzyme that transfers transfers it.
To transfer that, but you are not doing that. What you're
Just adding the
doing,
product, right?
you're adding the product of the faulty enzyme, okay? Okay, last one. If enzyme C is not working, all right, so now you face yourself some other way. There you go, okay? So now here we have a Brennan cell, neurosporal cell, exposed to X-ray, mutated gene C, and now there is a faulty enzyme C, not functional, right? I put this immunomedia precursor, what happened?
I take it and I make ortholine.
I take ortholine, I make citrulline, but I don't have nobody to pass it to.
That's right, so what happens to the cell?
The cell dies.
The cell dies. So what do I need to do in order to make the cell survive?
Add arginine, synthetic arginine.
Arginine, so I add the product of the faulty enzyme, arginine, and now the cell is able to live. Thank you. Yes.
One last,
Mhm.
you're gonna add the adenine without this enzyme C, but
Without the enzyme, yes.
without the enzyme. As the enzyme already created the adenine before,
Mhm.
as the enzyme, like, since he's dead, since he's not functional, how are you gonna get the enzyme, the adenine from him?
No, you're not getting it from him. You add the ingredient, okay?
Oh, you,
The researchers add it.
It's to check to make sure if it's functional or not.
That's right, that's what you're doing. You are not trying to replace the enzyme per se to add like a functional enzyme, but you are adding the ingredient to check which enzyme is the one that is faulty.
Because it's like a factory line.
It's a factory line, yes, there
Check
you go.
What robot's working, which robot's
Yes, yes. Actually, I was going to do like another example like that. Think about it. If, let's say, now here, yes, there you go. Let's hear. If you want to cook an omelette, right? You grab an egg and you break it, right? And then she puts it into the pan. Right? Put the whatever you put, right? The scramble it, and then you put it into the pan. Once it's in the pan in the fire, then it gets cooked, right? And then it lives, it grows, whatever, right? So,
On the
if
pan?
no, you are grabbing the egg and breaking it.
Okay.
Yes, if she's not working, there is no one to grab the egg and to break it, right? So what do? I just give them a broken egg so now she can put it in the pan and continue, right? And if she's not working, I just give him the pan with the scrambled egg already so he can put it in the fire, okay? Factory line, okay? So I'm just adding what they're supposed to be doing, the faulty enzyme, to check which one is the one that is not working, okay? Question?
Your
Does that-
cogs in a machine.
Does that help over there?
Your cogs in a machine.
Yeah, I just have a question, so I know the gene and enzyme, whatever.
I'll tell you later.
The precursor, the.
Factory
ornithine,
workers.
What do you call it?
Factory workers.
Yeah, the precursor, the ornithine,
Oh,
the rest
proteins.
Those will be like the ingredients, those are the precursors for the amino acid.
Yeah, I
Oh,
get it.
yeah, the
Call them as ingredients, you add in the ingredients to make the amino acid, the essential.
The amino acid is the adenine, right?